Welcome back to our series on statistical methods. I'm Mark Ledbetter. This is lecture 27, and we are going to discuss the code for chapter 14, which are nested designs. And we are only going to do two-way nested design, or two-level, one single nested design within the upper level. So the general form is a lot more complicated and would require a lot of modifications to this code. All right. So again, please do not change anything below date here. So of course you can change the date and your name and the title, please do. And then I'm going to clear everything so that this will be clear all outputs. So we'll have something exactly like, so this is exactly like what you will get. Okay. So what we need to do is run our chunk, our first chunk, and then our second chunks of, and we have some different libraries in here, and we have our table format that's required for later. Now this is not the RCBD example, this is a nested design example, and these are nitrogen content of sprays. Okay, so it's the same example we went over in our notes. So this example is taken from, excuse me, Applied Statistics and Analysis of Variance and Regression, second edition by Dunn and Clark, 1987. And I found the data on the internet and the description. So here is the description. I will not read it. I'll let you read it for yourself. That will keep this video shorter. And I've said what the first, second, third, and fourth columns are. And we have A equals 3 treatments or sprays, B equals 4 trees nested within each spray, N equals 6 leaves from each tree nested within each spray. And the order in which the trees are tested within each spray is random, and the order of the sprays is random as well. We do not, in this case, have the order that the observations were taken. So we will not be able to test independence. Here's the data. And it's very important that you make your factors correct. We said that the response is the nitrogen, so do not make nitrogen a factor or you will end up with garbage. But you do need to make you need to make spray into a factor because it's not numerical. It's 1, 2, or 3. You can see the first column here. And then you can see 1, 2, 3, and 4 repeated in the second column. That's your trees. They are not the same tree, obviously, within the different sprays. So within each spray, we have 4 different trees. And then we have 6 leaves on each of those 4 trees. So those, of course, are 6 different leaves. So leaf 1 from tree 3 is not the same as leaf 1 from tree 4. In fact, and tree 4 from treatment of spray 3 is not the same as tree 4 from spray 2. So these are all different. They are nested within each other. Okay. So the factor has to be the spray and the tree, and make sure that this here matches up with what the variable you put here, because when you don't do that, you make a very different model that's not any good. All right. So I took out the code about ordering this. If you need that, then you can copy it from one of the other codes that allows us. I think I'll put it back in here for your version, but comment it out so that you can put it back if you do get the order. We need to change A equals 3, B equals 4, and N equals 6 each time, and then capital N will be N row, but I don't think we use that. So of course you have to change your title here, and so we'll run this chunk and get a very nice table. And then we're going to do descriptive statistics. And so I've done this two ways. I've done the descriptive statistics just for the spray, which is what we're really interested in. We're wanting to know the difference in the nitrogen contents on the sprays. We're not really interested too much in the sprays within the tree, or trees within each spray. So and it's interesting that we need to reverse the order here to get it to have it grouped correctly. So I'll change that for you. So now when we look at this spray, you see spray 1 is all together, it's the second number here. So the 4 trees within spray 1. So that's how we would look at this. Now, what you'll see in my analysis is that I look at the medians for each of the main effects, the sprays. I look at the means. I tell you which the highest mean and median are with which one, the lowest mean and median are with which one, and the standard deviations go from the lowest, I say which one, to the highest, and then I only say that the means and medians within each tree are provided in the next table. Okay, so this will be table 3 here, which is here. But I'm not going to get into a big analysis of this, okay? And I don't expect you to either. Then we look at box plots. So with the box plots, I'm looking for, again, we're just going to look at what we're interested in, which is the main effect, which is the spray. And so what I want you to say is look at the bold line in the middle, the median. So it's lowest for number 3, it's highest for number 2, and then we look at the spread and the type of distribution. This is approximately symmetric. This is skewed to the left because the bottom is bigger. This is skewed to the left because the bottom part of the box is bigger. And so we need to say that. And so that's what I've said here. And I've said that the variation for spray 1 is largest and spray 3 is the smallest. So that's the size of the box, the height of the box. We've got dot plots. Again, I don't think these are the best way to look at this model, but I've included it. If I don't ask for it in the problem, or if you're doing a project, you probably do not want to include this, so delete this portion. And I've put the analysis for you here. There's not much I can say about it. It doesn't tell me a lot, unfortunately. And then we look at the mixed effects ANOVA. So I had to use some nesting. I've given you some information here. I hope you'll read this. I will not put it in your file that I give you, so please pause and read these 2 paragraphs here. Okay? And this way, this code is automatic. You don't have to modify it, so it updates itself. So we'll run this, and we're going to get the ANOVA table. You can see down here the ANOVA table, that so we can use this here in the console to write what we need to here. Okay, and so we have gone through and looked at we're testing, because we have a fixed effect for the spray, we do tau. But for the trees, they are random effects. They are randomly selected within each spray. And so they are a random effect. So together we have a mixed effects model. So for the effect of trees nested within A's here, within the sprays here, we're testing whether the variance is equal to 0. And that tells us whether there's an effect due to the trees or a difference in variation between the trees within each spray. Okay. And so we have the results written up exactly as we have before. Make sure that you change your the names of the variables to match what's being said. So, and again, here's our ANOVA that matches up with what we did in the notes. And here is a section for the various variance estimates and the confidence intervals for these variance estimates. Okay, so again, these should be automatic. There's no, you shouldn't have to update these or do anything with it. We run this chunk and we can see the output here. We get sigma squared. I said sigma b, but it's really sigma b within A. I guess I could change that to b a, and let's see. Okay, yep, so maybe, and I need to change this one as well. B within A. There we go. And so I've given the confidence intervals for all of these, so I've interpreted them. So you need to do the same thing. And if you were testing for whether these are significantly different than 0, which they all are, they do not contain 0 in the confidence interval, and they don't get very close either. So please read through this and make sure that you understand these estimates and confidence intervals. Now we go to model adequacy verification. So one of the things I need to point out is that here we used a linear model function, not the ANOVA, until here. Here's the ANOVA mixed one. And then we had to change some things because it's not a fixed effect. So if it's a fully fixed effects model, then you would comment this out here. Or actually, you could just comment this out so that you don't change it and then display mixed one, and it would give you the fixed effects ANOVA table. But as it is, it prints out, if you don't do this modification that I've got here, then it will print out the fixed effects. So it's going to take this mean square, and instead of dividing it by the mean square for spray within tree, it's going to divide it by the error. So it gives you pretending like it's fixed effects without any without it being nested. What it is, it's not just fixed effects, it's that it's not nested. So actually I need to take this out. That is not right. So this is for this is the mixed effects model, and it's for a nested model. So we have to modify this, or else it gives us the regular ANOVA, which assumes that the spray is not, or the tree is not nested within the spray. All right. So I've given you the confidence intervals there, and we do this exactly the same way. We test these assumptions exactly the same way. So, but here I have to plot the linear model, not the mixed. So it's a little different, but you don't have to modify this. So when we run this, we get the same 4 plots. Notice that I have an extreme outlier here. Okay, it's very far away from the line compared to all the others. That's going to cause some problems. And you can see that observation 9 is showing up as very far away from everything else in all the 4 plots. So this is going to create some issues for us. So let's take a look and see what kind of issue it creates. So when I'm looking at the residual versus fitted plots, I don't see any funnel shape. There's no one fitted value that has an obviously different spread, although the second fitted value has an extreme point. So by that I mean that this is the second fitted value, and 9 is an extreme point. Okay, it's further away than all the others, obviously. And when we go to the scale location, the red line is approximately straight and centered at 0 for the residual versus fitted plot. For the scale location, the red line is approximately flat and centered near 1, and there's no obvious funnel shape or other pattern. We have to not consider observation 9 here because it is obviously different than all the others. And if we look at everything without looking at it, if we ignore observation 9, everything looks fine. And then the same is true over here. Everything looks just fine within observation 9. And we can't look, this tree here is not the same as this tree. See, we have the sprays 1, 2, and 3, and tree 1, 2, 3, 4 within there. But tree 1 here is not the same as tree 1 here, so we can't compare those. All we can do is compare the trees within the spray. And we don't see a whole lot of difference there. Nothing that's really obvious for us. And so we come back up to that, and then we look at, say, the leverage plot. There's no obvious pattern or structure. We do not see a dramatic change in the edge points. No one treatment or spray has a significantly different spread than the others. We do not see a large variation between trees within each spray. And then if you want to do Levene's test, there's going to be a problem. Remember that Levene's test requires normality. And that big outlier is going to cause us a problem with normality. And so when we look at Levine's test, the first one for just between the sprays, it fails miserably. But again, this is no longer a good test or a valid test because we have the outlier. Now, within the trees, we see that there is no difference. So that's promising. But we did violate normality. Now let's take a look at the, oops, I thought I ran this. Let's take a look at the QQ plot with the 95% confidence bands, and you see that this is far outside. In order to even see this, we know that we should see a point that's quite far away from the rest. So when I did with -3 and 3, it's not even on the chart. So it looks like everything's just fine, but that point's missing. So you have to be very careful and make sure that you make these limits large enough. And then we see that, yes, we do have a point that's quite a bit out of the confidence band there, quite a ways away from the confidence band, not just the line. It's not borderline at all. It's definitely an outlier. Okay? And so, well, before we go back, let's take a look at the formal normality test. Again, this is going to be a problem. It's going to fail the formal normality test because we have that big outlier. But when we look at this, we see that this outlier is at the tail, and we're not concerned about it. But it's going to mess up all formal tests. The formal test for the equality of variances and the formal test for the normality. So they should, if you have an outlier like this, do not include those tests because they will not be good and you don't want to have to explain them away. People will say, well, why? Why did you include the test if you're not going to use it? So that is my advice to you, that I would never do the formal Levine's test here if I have an outlier. I've done both of them. Please read through them, make sure you understand. But you see, I said the failure of Levine's test is due to one outlier which skews the test results. So I would have, at this point, I would remove this sentence and I would delete this and not include that in the analysis. And then for the normality, most of the points lie close to the line. One observation, number 9, is far away from the line. Since it's at the tail of the distribution, we are not concerned with this one outlier. If we want to see if it's outside of the normality limits for 95%, we do that plot, which I've shown you previously. And we see that we have 1 point outside the confidence band at the upper tail. At this point, I would not actually perform the formal normality test because it's so far out, it's going to fail. And that's exactly what we saw, that the test for normality had a p-value, of I think 0.0010, so too small. Okay. But again, the note that the one outlier will cause a test of normality to fail is not robust. None of them are robust against outliers. Hence, there's no evidence that the assumption of normality has been violated. Our model is actually okay. And since we don't have the random order, we can't test the independence. So we conclude that the model assumptions actually have not been violated, even though we have that one outlier. This is very important. That's why everyone uses these plots instead of formal tests, because you see one issue, that doesn't negate everything that you're doing. It doesn't mean that what we're doing is not valid. We just have one extreme point. And since it's at the tail, we can expect that it might be extreme. Okay? But it's not going to cause us to throw out our results or to question whether they're valid. Okay? So now we have Sheffey's test. Okay? So now, Sheffey's method, and so when we run this, we only need to do it by the spray. It's the only fixed effects. Do not run it for the trees because the trees are random effects and it doesn't make any sense. We don't do contrast for random effects, only for fixed effects. So, so make sure that you read through that. And then I've put this as a test of tau 1, tau 2. What I could really do is say that this is mu I minus, because these are these are really, this is not the right test. I should change this. Mu j versus mu I minus mu j are not equal. Not equal to 0 for at least 1i not equal to j. And okay. And then we have to do, and so we have the experiment-wise error. In this case, I've set it at 0.05. And when we run this, let's see, yep, okay, so there they are down here. We don't have 0 in any of the confidence intervals, so we know that that they are different, okay? All three. Notice that 3 and 2 are actually closer to each other than 2 and 1 or I'm sorry, 3 and 1 are actually closer to each other than 3 and 2 or 2 and 1. Okay, because if you go back to the box plots, you'll see that those are closer together. The closest two values together are 3 and 1. Okay, for the means. Okay, so here I have written this up. Please read it and make sure that you understand how I am doing this. And then we go to Tukey's method, and we have, again, instead of tau, well, this should be mu I minus mu j is equal to 0 versus they are not equal to 0. The difference is not equal to 0 for at least 1i not equal to j. Okay. In the model, experiment-wise error in the model. So here I've written this up. Now, this gives us p-values, so we can do the hypothesis test. And yeah, I could also do the confidence interval here. I didn't do it for each one of these, but you could copy from down here where I've done the confidence interval. You could copy this and paste it here and do the confidence interval and just change these values in the confidence interval. Okay, so even though I didn't provide it, you can still do it. So I've written it up. Now, if you're asked to do a hypothesis test, this is how you write it up. If you're asked to do a, and so I've written these up. If you're asked to do a confidence interval interpretation, this is how they're written up. Okay. Now we're going to go to the custom contrast. I've again just done the pairs. So there is a limitation here that you can only do a minus 1 contrast with this method. So even though I have 3 here, I only use 2 of them. Okay, and so if I use 2 of them, I need to make sure that I change M. Even though I have 3 listed, I only use C1 and C2 here because it won't let me use 3 of them. It will give me an error. It doesn't. This function won't let you do that. It says too many. Okay, so so you choose the 2 you want. I could change this to C1 and C3. And if A was 4 and I wanted to use all 3 of these, I would change M to 3 and I'd change N rows here to 3 because and then I would add C3 here. Okay, so the number of objects here has to match up with the number of rows here. And then if I have 3 of these, I would need to add another name here for that third row. Okay, but so that was only if we have more. All right, so in order to get this to work, I had to create another object here. It didn't like my other models. And so again, because I've changed in row to 2, I have to change the number of repeats here to 2 as well. These numbers here within this REP function, the number of times it's repeated, this has to match the number of rows here, which matches how many of these we have, which would also, if you're using Bonferroni, match M. And again, I've said if you want Fisher LSD, change M to 1. Okay. I'm making this the Bonferroni by dividing by 2 for these. Okay. Now, I did not calculate a p-value for this. So how do I write up a hypothesis test? Well, if the interval, which is here, does not contain 0, and this is a 95% confidence interval, then the p-value is less than 0.05. That's all I can say. If 0 is in the confidence interval, then the p-value is greater than 0.05. So the only thing you can do with the p-value for this one is say it's less than 0.05 or greater than 0.05. And so I've written up both of these here as you would for a hypothesis test, and then again for the confidence intervals interpretations down here. Okay, so I hope this helps you do your homework and the projects, and so please take care of yourself, stay safe, because we hope to see you here next time.